I'm Kira Curtin, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Cloud Native Single Cell Data Management, powering the largest public single cell repository in the world, and is sponsored by TileDB. Our speakers today are Pablo García-Níeto, a computational biologist at the Chan Zuckerberg Initiative, and Aaron Wolin, Principal Software Engineer and Project Leader for Single Cell at TileDB. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Pablo. Pablo?
Thank you, Kira. Hello, everyone. As Kira said, I am a computational biologist at the Chan Zuckerberg Initiative, or CCI for short. And I'm here to present to you today about a new recently launched feature of CELLxGENE Discover called Census. But before I give you more details about CELLxGENE and Census specifically, I wanted to set the stage for what we're dealing with in the single cell space. In particular, at CCI, we have a 10-year mission to support the understanding of the mysteries of the cell through measuring human biology. And to that end, we have a single cell team at CCI. And in the team, we aim to further this understanding of the human body and diseases at the most fundamental level unit of life, which is the cell. We do this by accelerating single cell biology methods, technologies, and applications. But why single cell? Well, humans have in the order of tens of trillions of cells, and the genome encodes for more than 60,000 genes. Single cell technologies give us the highest resolution of what's going on inside a single cell at any given point in time. And we can get this resolution because with these technologies, we can obtain gene expression matrices for all of the genes in the human genome across hundreds of thousands or millions of cells within any given sample that we are studying. And we can start analyzing this data as a whole to really get to what is the cellular function of any given cell in the human body, what are the underlying causes for a specific disease, and also how these cells work together as a system to enable body functions. And importantly, start to differentiate that genotype versus environment aspect of biology at a very high resolution. Because of this, over the last 10 years, there has been a huge increase of the number of single cell studies that get published every year. And in particular, over the last few years, we have seen in the order of hundreds of single cell studies that get published at any given year. Now, these studies, they are focusing on a specific biological context, and they go really in depth about a specific question or context that they may have in mind. But now more than ever, scientists are very eager to start compiling all of this data and perform more system-level analysis that will get us closer to an understanding of how the human body works as a whole system rather than individual modules. A clear example of this is a paper that was published earlier this year by Christina Theodoris and others, where they developed GeneFormer. So GeneFormer is a language model, which is a context-aware, attention-based deep model, and it was trained on 30 million cells. So once you have these type of foundation models, you can use them for a bunch of different tasks. One very good example that they provided was, given these disease cells that I have, small set of disease cells, what are the top candidate genes that when I change them up and down, I can go from a disease state into a healthy state? So we can ask these type of questions to the model, and they provided an amazing example of this in a specific cardiovascular disease. And they also provided experimental validation of the top candidate genes that they found to go from that disease state into a healthy state in an in vitro system. And the results look amazing. So I'm very excited to see where this goes into the clinic. But now, to enable these type of applications, these researchers had to go through the very laborious process of getting all the data out of the literature, standardizing the data, putting it in the right format, doing vocabulary conversions between the different data sets to be able to get to this state of modeling. So this is the problem at scale, the problem at large. We have a scale problem. So there is continuously new data that gets generated, and individually it's large, but in aggregation it's even greater. So it's very hard to download or copy or move around. A great example of this is what we currently have in the cell by gene data corpus, where we host tens of millions of cells, and it's really large to really provide a scalable access to this data. And the other big problem that the community has currently is that there's a lot of language-based data silos. So we have mainly the Python and the R communities, and they provide incredible algorithms that support analysis of this data. But more often than not, these algorithms only exist in one language or the other. And it's very difficult, almost impossible, to be jumping between the two of them to perform an entire workflow of analysis. So at Single Cell, at CCI, the Single Cell Tech team, we have two main aims. We want to democratize access to single cell data for all experimental biologists. So the single cell field is still a bit niche, and we want to increase access to everyone that's out there. And in particular, anyone who wants to understand human biology. To do this, we need to have reusable data and processes with user-friendly interfaces. So we're putting a lot of effort into that. And on the other hand, we need to foster computationally based applications in the single cell space, like the one I just showed you before. So for this, we are working on increasing efficient access to this type of data, having high quality harmonized data, and then providing all the tooling necessary to perform analysis and modeling at scale. Now I'm going to pass it to Erin, who's going to give you a quick introduction about how TileDB is trying to address these big issues in the data space, but specifically in the single cell space. So it's all yours, Erin.
Great. Thank you, Pablo. So I am going to talk about SOMA, which is the open source project that we've been working on in collaboration. Presenting single cell data on disk and an API specification for interacting with that data model. Both of these are open source and language independent. For SOMA early on in the project. First, of course, it needed to be open source to encourage community adoption, feedback, and contributions. It should be language independent to avoid some of the data silo issues that Pablo just referenced that crop up when you're using these framework-specific formats. It needs to be interoperable with the popular single cell analysis framework so users can store their data once and analyze it from anywhere without having to convert back and forth between file formats. And of course, it needs to be able to scale to meet the demands of census scale data sets like the one CZI has produced. And this requires features like support for out-of-core reads and writes, efficient slicing and filtering, and it needs to be cloud native so it's possible to take advantage of cheap, highly available cloud storage. And as you might have guessed, TileDB was chosen as the underlying technology for SOMA. If you're not familiar with us, we are a data management company that builds integrated solutions on top of our core technology, TileDB, which is a highly flexible multimodal database built around n-dimensional arrays. The core library is open source and MIT licensed, and it's used for a huge variety of use cases. Just within the life sciences domain, we have customers using TileDB for biomedical imaging, population genomics, and of course, single cell data management. So whether you're working with tables, arrays, genomic variants, time series data, they can all be stored in TileDB and then queried using a common set of APIs that we've built for many different languages and frameworks. So why was TileDB chosen? So I'm going to go over some of the features that made TileDB an ideal choice for SOMA, and by extension, the CZI census. So I already alluded to its flexibility in terms of being able to model many different categories of data, but this flexibility also extends to its indexing and compression capabilities. TileDB array dimensions are indexed for fast query performance and can use a variety of data types, including ints, floats, dates, times, and even arbitrary strings. So again, whatever the use case, TileDB can model it effectively. And we leverage many of these capabilities in SOMA because a typical single cell data set will comprise both sparse and dense data, as well as tables and metadata. And of course, scalability is going to be one of the most pressing challenges that people are facing across all domains, really. And that manifests in a couple of different ways, including storage capacity, I/O performance, maintaining query performance as data sets grow in size. And TileDB was designed with all of these challenges in mind. At the lowest level, it uses a technique to split data into smaller, equally sized chunks, or tiles, as it were. So when a query is performed, only the relevant tiles are loaded into memory. It's also a columnar format, which means you can efficiently compress data within each tile to produce a very compact representation on disk. You also have the flexibility to choose which attributes from each array should be included in the result. So if you're working with a large data set and you only need a few of the columns, you can just select those and ignore everything else. TileDB is also cloud native, and it's highly optimized for working with cloud object stores. So when you write data into a TileDB array, you can pass it a local file path. You can also pass it a URI that points to an S3 bucket, and it just works. There's no additional software required. And obviously, this was a critical consideration for the census, so they can economically store this massive corpus of data and make it available to the community. And of course, it's highly performant. So the core engine is written entirely in C++. All read and write operations are fully parallelized. So you can take advantage of all the cores on your machine or server. It also supports multi-reader, multi-writer concurrency, which allows you to have multiple processes reading and writing to the same array at the same time. And because the core library is all C++, we can easily build on top of it to provide APIs for many different languages. So you can use TileDB from Python, R, C#, Java, and others, Go, and still some others that I can't think of right now, but they're all built on top of the same core library. So you always get the same performance no matter which of these you're working with. So now let's SOMA's data model, which should look semi-familiar to anyone who's worked with any of the popular single cell frameworks we've mentioned earlier, because there are a lot of commonalities. And I should point out that while I'm saying single cell data over and over, SOMA is really designed to be a general purpose data model for representing any type of annotated matrix-like data, which has a lot of applications in life sciences. And really, it's the bioconductor folks who did a lot of the pioneering work in this domain, figuring out how to represent and interact with this type of data easily back in the microarray days. And since then, we've seen really nice refinements and extensions from packages like ANData, Seurat, and many of bioconductor's modern packages like single cell experiments. And we have incorporated a lot of the lessons from these packages into SOMA. We also adopted some of ANData's semantics. So if you're familiar with that, you will feel right at home working with SOMA. But there are a few key structural differences that I will point out. So let's go through it. The primary data structure for a data set in SOMA is the SOMA experiment, which is a multimodal container that comprises two components: a data frame called OBS that stores cell annotations and provides an index for the cells in the data set, and MS, which is a collection of one or more SOMA measurements. SOMA measurement is a container for a single modality of data. So if your data set includes multiple assays like RNA and protein, the SOMA experiment would contain two SOMA measurements. modal containers for the actual data. At minimum, they contain a data frame called var that stores the feature annotations and provide an index for the features in that data set, and then a collection called X that contains one or more arrays with the actual assay data. And we refer to each of these arrays as a layer. So one X layer may contain the raw counts, another may contain the normalized counts, and so on. Similar to AND-data, we provide additional collections for storing the derived results. So the obsm and varm collections are for matrix-like data with rows that map to the cells or features, respectively. So you can use these to store things like your dimensionality reduction results. And then we have obs storing pairwise relationships between cells or features. And you can use these to store things like cell-cell adjacency matrices. So now that we've finished the data model of the API, and let me just check my mic here. I just got a note that I'm going in and out of mute. So let me switch mics, see if that helps. Okay. All right, let's try again. So now we're looking at an API. At the API for SOMA, we'll look at some sample code to see how you can quickly get started with it using your own data. As I mentioned earlier, interoperability with popular single-cell frameworks was a major priority for SOMA. So we provide I/O methods for the most commonly used single-cell data formats. In this case here, we're using an H5AD file that was downloaded from the Cell by Gene data portal. And we're going to use the SOMA Python API to ingest this data into a new SOMA experiment located on S3. We just need to provide a name for the SOMA measurement that will contain the assay data. So we're calling that RNA here. And then give it a URI to determine where the SOMA experiment will live. And like I mentioned before, this could be a local file path, or it could be an S3 bucket. In this case, we're using a special TileDB cloud URI that will create the new SOMA experiment on S3 and then automatically register it in TileDB Cloud's data catalog. Once your data is ingested, you can leverage SOMA's powerful query interface, which provides a lot of flexibility for slicing and filtering your data. In this case, I'm performing the same query in both Python and R, which is simply filtering for all cells that have been annotated as platelets. But you can make these queries as complex as you need to by combining multiple filters across both cells and features simultaneously. An important point is when you run this query, it only identifies and returns the relevant indices from TileDB. No other data is loaded into memory until it's explicitly requested, which is a key feature for working with large data sets. So after performing the query, we can inspect the results to see how many cells and features matched our query conditions. We can then selectively load any part of this experiment into memory, or you could use the toAndData method to read the query results into memory as an AndData object. And notice we can even sub-select which attribute should be included in the result. So again, you have a lot of flexibility here. And of course, we provide similar methods for working with Seurat and bioconductor objects if you're using R. So again, this allows you to store your data once as a SOMA experiment, and then gives you the flexibility to analyze it with Seurat, bioconductor, or ScanPy. And if you work in an organization that's looking for a solution to manage and analyze your single-cell data at scale, I wanted to briefly mention TileDB Cloud, which is our commercial SaaS product. Everything I've shown so far is available in the open source SOMA library, but TileDB Cloud provides a lot of additional functionality for organizations, such as a built-in data catalog for organizing your data and making it discoverable, access controls and logging, simple point-and-click sharing of data, code, and files, hosted Jupyter notebooks and dashboards, as well as a serverless compute platform for easily scaling your analyses and pipelines. And as a quick example, here's what that data set we ingested earlier looks like in TileDB Cloud's UI. So every asset gets a nice landing page like this, which allows you to interactively explore the structure, examine the schema or any associated metadata. You can share it right from here with other users or organizations. And you can even make something publicly accessible, and everything is logged. So you can always see who accessed what and when. On the right, we are looking at a hosted Jupyter notebook, which is running on a TileDB Cloud instance. And in the notebook, we've executed a serverless pipeline that's applying the same query to each tissue-specific SOMA experiment from the Tabula sapiens dataset. Each query is performed in parallel in its own node, which is automatically spun up and down on demand, so you only pay for exactly the compute you need. And this is a admittedly simple example, but we offer a very powerful API for building out complex workflows that work very well with SOMA and SOMA datasets. So with that, I am going to hand it to talk more about the amazing resources that CCI has built using TileDB and SOMA. Over to you, Pablo.
Thank you, Aaron. So I will give you a quick introduction about what CELLxGENE Discover is, and then I'll jump into the census feature. So CELLxGENE Discover, it's an online platform for go right back at my slides. There you go. I think somebody's moving unintentionally my slides, so going back one more time. There you go. CELLxGENE Discover is an online platform for browsing and analyzing standardized single-cell datasets, explore gene expression, or just download the data to have it at your fingertips. The standardized aspect of it is very important, and we put a lot of effort into it. So currently, we have over 45 million unique cells at CELLxGENE, and these all come from more than 800 different datasets. And even though we have been focusing on building a reference healthy data, we are now including in the order of 80 diseases in the corpus. And you can see on the right side of this slide that our ingestion rate of new data, it's linear and constant. So we don't see these numbers being slowed down anytime soon. And actually, these numbers that I'm showing you right here were taken about, I think, a month ago, and currently we went over the 50 million unique cell mark. So we're very excited to keep building on top of this resource to have the most comprehensive standardized single cell dataset of the human body. So coming back to the standardization aspect of this, every single dataset that we bring into CELLxGENE Census has to have and has to follow a specific data and metadata schema. So all of the data that comes into CELLxGENE has to come with row counts, and we allow normalized data by the contributors so long that they provide the row counts as well. The cell type is standardized on the variables that I'm showing you here, and for some of them, we have ontologies that we use to understand the relationship between values of these variables. So for example, we use the cell ontology to encode cell type, and that way you can very easily understand the relationship between different cell types within our data corpus. And then for feature metadata, currently we use a PIN version of the gene code genome version to standardize on the names and the IDs of our genes, and then looking forward, when we start supporting protein data, we will start looking into how to best standardize that feature space. So we have a few different visual tools that are actually powered by TileDB. We saw the opportunity that TileDB offers to scale and to provide tools that work at a very interactive speed. at very large scales. So these two tools on the top here, one is devoted for analyzing single data sets at any given time. So this is a UMAP visualization tool or scatter visualization tool, where you can interactively take a look at the different cell metadata, gene expression, and then perform slices of these data using the browser. And then we also have gene expression along with cell guides, which was recently launched also, that leverage the full data corpus that we have at CELLxGENE to provide views of gene expression, as well as all the cell types that we currently host in CELLxGENE. And finally, we have very easy ways to browse and download any data set of interest for you using interactive visual tools for that. But now I want to do a deep dive into Census. So Census is a feature that was built for computational biologists, and it is both a TileDB-SOMA data object plus some Python and R APIs that sit on top of TileDB-SOMA to provide efficient, free access to all of the data that we have in CELLxGENE using a cell-based paradigm instead of a dataset-based paradigm. So the way we built Census was by taking all of the standardized datasets that we have in CELLxGENE, concatenating them into a large TileDB-SOMA object that contains all the more than 30 million unique cells for human and around 5 million unique cells for mouse across all the 60,000 genes in their genomes. We also provide the cell and gene metadata tables for every single cell and every single gene in this large object. And thanks to TileDB-SOMA, you can query and slice Census any way you want, either using coordinates for cells and genes, but more importantly, using value filters for specific values of the standardized cell metadata and gene metadata that we have. And again, thanks to TileDB-SOMA, you can stream and export the data into some of the most common single-cell toolkits in the ecosystem right now, both in R and in Python. But also, more importantly, you can interact with the data directly via TileDB-SOMA to start performing analysis at scale within reasonable compute systems. So I just want to touch base on some details of the data that we include in Census. So from all the data that we have in CELLxGENE, we only include data in Census from cells coming from mouse or human. We currently do not include spatial data, so only RNA non-spatial data. And we only currently provide row counts, although we recently just added a normalized layer, and I'll go into those details in a second. And we also only include the standardized cell and gene metadata from CELLxGENE Discover. A small note that is important here, we combine into Census data that comes from full gene sequencing read counts and also molecule counts. So there's a combination of SMART-seq data as well as data, for example, from 10x and other technologies that actually do molecule counts. There's also some level of duplication in Census because there are meta-analyses in CELLxGENE Discover, and then within one collection, for example, Tabula Sapiens, we can have the data divided into different data sets. So there's some level of duplication there, but we allow you to identify what we call the unique cells by this cell metadata variable called isPrimaryData. So Erin gave an amazing introduction as to the different type of objects for TileDB-SOMA, and this is a demonstration of how we're using the TileDB-SOMA specification within the context of Census. So this is what the Census object looks like. We have two large collections, one that is very general purpose, and it demonstrates the flexibility of TileDB-SOMA because this does not contain any single cell data whatsoever. This just contains summary tables as to general information for any given version of Census. So for example, it contains a table of all of the data sets that were used to build this version of Census. And then we have a collection with the census data, and this has the single cell data hosted in there. So we have two different SOMA experiments, one for Homo sapiens and one for Mus musculus. And within these two different experiments, we have one single measurement right now for RNA data, and then two layers currently, raw and normalized data. The normalized layer is just normalized by library size across all cells in Census. And we have the expression matrix along with the axis data frames for gene and cell metadata, but we also have a specific matrix that lets you identify which genes were actually measured in different cells. We are planning on publishing long-term supported census releases every 6 months. And we promise you that we're going to make them available for up to 5 years upon publication. And to keep up with the speed of data ingestion at CELLxGENE Discover, we're also going to publish weekly releases every Monday, and they will be available for up to 1 month after publication. So the weekly releases help you stay up to date with the latest data, and the long-term supported releases help you create reproducible results in your analysis pipelines. All of this, again, thanks to TileDB-SOMA, is hosted in Amazon S3. It works out of the box, and it's free of use, free of charge for anyone who wants to come and play with the data. Now I'm going to jump into a few examples of how this looks when you're actually using the APIs. So here I have an example for the Python API. We're loading the CELLxGENE Census API, which also loads the TileDB-SOMA API to work on the census. We provide very convenient functions to open a handle to the S3 bucket. So by calling this method openSOMA, you get a direct handle to the census TileDB-SOMA object that's hosted in S3. And using this TileDB-SOMA object, you can quickly start performing queries on the data that we have. So in this example, I am showing you how to read the ops data frame that contains the cell metadata, but I'm only selecting specific cells of interest. So I will select only female cells from microglial or neuron cell types, and I only want specific columns of this data frame. So assay, cell type, tissue, tissue general, suspension type, and disease. As Erin mentioned, nothing really gets extremely downloaded here. So we just make a connection to S3, perform the query, but we don't download the data yet. We get an iterator of PyArrow tables that we can concatenate very easily to have a large PyArrow table, and then we can convert that large PyArrow table into a pandas data frame that we can start using for any downstream analysis. So this entire process of the query and the data download takes in the order of 30 seconds with a good internet connection. And at the end, we get a data frame with 300,000 cells coming from multiple different data sets. And now we have them at our fingertips to start analyzing. This is another example that gets to the point of interoperability. We have equivalent examples for R, but I'm just currently showing you the Python examples, where we can very quickly and easily, following the same querying capabilities, we can export any slice of census into AND data to be used for ScanPy and other tools that work natively on AND data. So very similarly to the previous example, I am opening a handle to the online object, and I am selecting one of the two different organisms that we have, Homo sapiens. And in this specific query, I will filter for two different genes, the same cells that I just filter on, and the same columns for the ops data frame. So within less than 1 minute, we can obtain the gene expression data for all of these cells across the two genes that I just selected in an AND data object, and I can start my analytical pipeline with this object. And I want to quickly touch base on what we believe is one of the most powerful aspects of TileDB-SOMA. So currently with the AND-data workflow, what's going on under the hood, we're downloading the data and we're loading everything into memory. But the power of TileDB-SOMA comes when we can interact with the data in sort of an incremental fashion to not load everything into memory. So to that end, we can perform an experiment query, which looks very similarly to the previous examples that I just showed you. In this case, I am selecting all of the cells from brain and male cells. I perform a lazy query. And after I perform this lazy query, I can obtain iterators for all of the different objects within the SOMA experiment. So I can obtain iterators for the X matrix, iterators for the axis data frames, both the cell and the gene metadata, and I can iterate over these iterators to obtain and stream small pieces of data at any given time that I can process incrementally. We already have 2 demonstrations of how we can use these capabilities to perform single cell specific analysis. So for example, we provide an API that lets you obtain the highly variable genes across the full human data in a regular laptop. And this works under 20 minutes as it's streaming the data, calculating average and variance accumulators to obtain the highly variable genes. And similarly, we provide incremental APIs for mean and variance calculation that similarly work in just a regular 8 GB memory laptop under less than 20 minutes for all the more than 30 million cells in the human data. I'm gonna switch gears into some latest developments that we've done on Census, which relates to machine learning. So we developed PyTorch loaders that utilize these TileDB-SOMA capabilities of reading data in chunks, in this case, to perform modeling at scale. So this, we developed a PyTorch data pipe, which works in our case very similar to the experiment access query that I was just showing you. And we put a lot of effort into creating a user-friendly API that you can start using right away. And so we call this the experiment data pipe. And it is a PyTorch iterable style data pipe. So very similarly to the get and data function, we can initiate one of these data pipes by selecting specifically the experiment that we want to work on. So either the Homo sapiens or the mus musculus experiment, the layer that we want to, or the measurement that we want to work on, as well as the layer of data that we want to retrieve data for modeling. We can specify, again, a slice of the cells that we want to work on. In this case, is tongue and only the unique cells. And then we have to specify what columns from the cell metadata data frame we want to utilize in case you're doing an inference workflow. So for example, I want to create a model that predicts cell types. So I could select the cell type cell metadata for us to model on that. And we can also specify the size of the batches. So this is just the PyTorch batch size that lets you know how many cells you will get in each iteration of the loader. Because this is a PyTorch experiment, a PyTorch data pipe, you get access to all of the functionalities that are available via PyTorch. So you can split your sets, you can shuffle your data, and then you can initiate the loaders as well. All of this happens in a lazy manner, and not only until you start performing the modeling, you start actually downloading the data and performing operations with that. And speaking of that, this is what the tensors look like for every batch that you get during the modeling phase of your analysis. So in this case, since we specify a batch size of 16 cells, our X tensor is going to look like a tuple of cells, and each cell, in this case 16 different cells, will have the gene expression values across all of the genes that we selected, in this case, across all 60,000 genes. And then we're going to get an ops tensor, which gives us the ID of every single cell that we can map back to census, and also the cell type encoding. Remember, we selected the cell type column from the ops data frame, and in this case, we encode those labels into integers, and we have encoders and decoders that work very well to get the labels out of these tensors as well. Alrighty, and so what's coming to census? In this year, we're working really hard to get you more data releases. I mentioned we're going to have every week a weekly release, and then in November, we're going to release the next long-term supported data release. We want to increase access efficiency to our data. So we want to provide data mirrors around the globe. The census is currently hosted on the west coast of the United States. But as Erin said, TileDB-SOMA is cloud friendly, and we can just very easily create mirrors around the globe to increase data access based on your location. We're actively looking for feedback on this. We're about to roll out mirrors on Europe, potentially Germany, and East Asia, because those are the regions of the world right now where we see, after the US, the highest usage of census. But if you have feedback for us, if you're going to become a power user of census and you'd like it to be close to your geographical location, please let us know. We're working in collaboration with TileDB to increase our data caching capabilities so that you don't have to be downloading the same data all the time when you're performing very similar queries. And this is very important for the PyTorch loaders. You want to have a way to cache the data, especially when you're going through several epochs in your modeling phase. And we actually already have full GPU support on the PyTorch loader. So this is something that we already accomplished. TileDB has worked very diligently to provide support for categorical variables. And we are in the middle of rolling out these changes into TileDB, TileDB-SOMA, and eventually into census. We recently added a normalized layer and some pre-calculated statistics. So number of non-zero values per cell, the sum of the values across all cells or all genes, and a few other pre-calculated statistics that you can actually use for filtering your data. And most importantly, one of the biggest aims for CCI currently is to provide integrated embeddings and the models associated to those integrated embeddings. So we're very actively working on this space to increase the usability of our data. And finally, we want to leverage more of the TileDB-SOMA capabilities on incremental calculation or incremental analytical tools in the single cell space. So we want to provide capabilities for differential gene expression, subsampling, marker gene discovery, and potentially also in the clustering space doing some KNN graph generation, as well as scalable PCA calculation. With that, I just want to thank the team at CCI, and in particular, the amazing team that has made the census happen over the last year. They're all highlighted here, so big kudos to the team. We're here to help. We're always looking for collaborations to help you in your research or any feedback that you may have for us. Feel free to email us at soma@chanzuckerberg.com. File any issues in our GitHub repo, and most importantly, come and join the Slack and go to the Cell by Gene Census users. So this QR code lets you go into the registration page for Slack. We're very actively looking at it. If there's any questions that come up, we're very quick to answer those questions. And finally, come and visit the census documentation page for full details on how to use the census. We have a bunch of different tutorials. Again, we're always eager to hear feedback. If there's something missing in the documentation, if something is not that clear, please let us know, because this is, again, a community-driven type of project where we're leveraging all the data that exists out there in the single cell space, and we want to make it available to all of you guys. So we really want to improve the way we can help you get started with using this tool. So I'm now going to pass it back to Chiara, who's going to take care of the Q&A. And thank you all for being here today. And I hope I hear back from some of you.
Thank you, Pablo. As a reminder to our webinar participants, if you have a question, please type it into the Q&A box that's in the control panel. And we'd also like to ask attendees to take a moment after the webinar is ended to take our exit survey and provide us with your feedback. And so we'll start with the Q&A portion now. And the first question is, kind of, to what extent does not having the spatial context or the information about where cells are embedded affect some of the possible conclusions from single cell profiling strategies or studies?
I'm going to try to take a stab at this question.
Okay.
Hopefully I understood it correctly. And yes, so context in which a cell was sequenced and sampled is very important. So one of the easiest example of this is the cell type or the tissue that is coming from, right? Or what technology it was used to sequence that data. And so with the cell by gene schema, we're trying to aim for a good balance between a simple yet comprehensive schema. So as you might have seen, we standardized on a few cell metadata variables, but we constantly hear from users, hey, what if you add this variable to your schema? This is an important context that we need to understand to do downstream analysis. And we stay very active in hearing these suggestions, and we incorporate them as we see able. So one example of this was suspension type. So differentiating between cell and nucleus sequencing. This is a recent addition that we did to our schema because we understand the generability and its use for the single cell community. We're always trying to improve in this regard, but there are some cases where it's very hard to have high generability for a specific metadata variable, and we tend to go towards the flexible simple schema in this case so that we can keep our ingestion of data at a very high rate. But yeah, thank you for your question. And if you have any specific variables that you'd be interested in including into our schema, again, we're always happy to hear those suggestions.
Okay, thank you. Another question is, if someone wanted to get their data into Census, how would they go about that? Who would they contact?
Sure. Feel free to email us either at soma@chanzuckerberg.com or at cellbygene@chanzuckerberg.com. We work with a group of curators because, as I said multiple times, standardization is a huge aspect of cell by gene. So we work with a team of curators that go over contributor data and help the contributor wrangle and standardize the data so that they can put it into Cell by Gene Discover. So usually the process goes, a contributor contacts us, we put it in the queue, and we start working with you to help you get the data into the portal.
And do you process the single cell data from raw FASTQ files?
Yeah, thank you. That's very important. We do not process the data from the raw files that come out of the sequencing machines. We were very intentional about not jumping into that space because it's just a different monster. And again, we wanted to get very quickly to a point where we have a very large compilation of standardized data. And so we asked our contributors to provide the simplest or the easiest representation for them of the process gene expression matrices along with the cell line gene metadata. Sometimes that comes in the form of a MTX file, for example, from Cell Ranger. Sometimes that comes in the form of an AND data or a Seurat. And we work again with this amazing group of curators to help you wrangle data and transform the formats into the formats that we accept for ingestion.
Do you know if there are any plans to integrate statistic packages or to provide pre-computed statistics on the cell census data?
We are slowly working on the compute capabilities and the statistic capabilities of TileDB-SOMA specifically in the context of census. As I mentioned, we've developed a few demonstrations of gene and variance calculation, highly variable genes, and now we're working into the modeling space. So yes, we're interested in that. I think it's the type of work that requires a lot of effort on our end. And again, we want to identify what are the highest impact areas where we can start working on.
And so the census is RNA-seq data, but do you have plans also to expand to other modalities like ATAC-seq or others?
Yeah, so we're currently actively working on trying to expand to spatial data. We wanted to pick our big next modality, and we were discussing between some of the spatial technologies that are out there, as well as some of the non-RNA technologies, for example, as you mentioned, ATAC-seq, but there's also DNA methylation sequencing out there. And after some consideration, we understood that the community is really eager to get their hands onto the spatial aspect of single-cell genomics or single-cell data. And so that's why we decided to work next on spatial. So hopefully by next year, we're going to have some exciting results and prototypes to show you guys.
Great. And is it possible to search the ontology by gene rather than by cell type?
It is possible to search by genes. We don't use a gene ontology. We just use the gene code vocabulary for gene names and gene IDs. So everything has ensemble IDs and their corresponding gene names. So that's as far as it goes. I'm not sure if maybe I'm missing something about that question on ontologies.
Okay, let's see if they follow up. Okay. It looks like you can already ingest and data files, but how can I ingest data in other formats to TileDB-SOMA?
I'll pass that to Erin.
Sure. So I touched on this a bit. Right now, SOMA provides I/O methods for H5AD files, and data, Seurat objects, and bioconductor single-cell experiments. Of course, there are additional formats that we very much would like to support in the future. And this is where community feedback is really important to us. So if there's a format that you're using regularly that you would like to see supported by way of an ingester or an exporter, please let us know by opening an issue on our TileDB-SOMA repo. Maybe one of our backend folks can paste that URL into the chat for everyone. But it's called TileDB-SOMA, and it lives in the single cell data organization on GitHub. But we take those kinds of feature requests very seriously, and they give us a sense of what other formats people are working with and what should be prioritized.
Okay. Going back to the ontology a little bit, have you gotten feedback from cell ontology curators on cell types that are poorly annotated? To maybe help guide community efforts to annotate and re-annotate cell types?
Yes, we are in constant chats with some of the cell ontology maintainers, as well as some of the EFO ontology maintainers. So EFO is the ontology that we use for encoding what sequencing technology was used for any given cell. And they're very open to all of our suggestions, and they're very collaborative with us. So anytime our curators find a new term that needs to be added to one of these ontologies, or a term needs a correction, it's very easy for us to push those fixes into the ontologies. I think the challenge comes more not evolving the ontology, because that seems to be working really well, but rather making sure that contributors are using the right term for their cell annotations. So it's happened a few times that, for example, somebody marks a cell as a progenitor cell when they actually meant to use a myeloid progenitor cell, which are two different cell types as defined in the cell ontology. And those type of annotation errors are very hard for us to catch. And again, this speaks more to the importance of our curation team. But some of these errors we don't catch until a user tells us, hey, your data doesn't look right. And we realize it's an annotation issue. And we try our best to correct those annotation issues.
Okay. And we've gotten a follow-up on that previous cell ontology question. And so they say that the question is specific to cell ontology, and which provides the marker genes for each cell type. And that the question is, if I have a gene, can I find if it is a marker gene for any of the known cell types?
Got it. This is probably speaking to some of the visual tools that we have in Cell by Gene, where you can actually explore all of the cell types for all tissues that we have in Cell by Gene. The tool is called Gene Expression. And then for any given cell type, you can click on the cell type and obtain the list of gene markers. And so the question is, how can I go from having one gene and see whether that's a marker on any of the cell types? We currently don't have a tool for that. Thanks for the feedback. What we do have is, or what we do plan is to put out there our table of gene markers for public use. And so you can quickly just download that table and start sort of like wrangling the table in a way that works for your analysis. So you'll be able to fulfill that case when we put that table for public use. But unfortunately, we currently don't have a visual interactive way to get to that question. I'll bring that back to the team. Thank you.
Is SOMA going to support SRAT version 5?
Yeah, so we are really excited about this new version of SRAT and the new functionality it brings in terms of transparently supporting on-disk data, as opposed to doing everything in memory as the previous versions do. Obviously, we think this is a great opportunity for SOMA. And in fact, Paul Hoffman, SRAT's lead maintainer, is now part of the SOMA team. And seamless integration with this new version of SRAT is very high on our priority list. So that's something we'll have some news to share about in the future.
All right. And can you summarize some of the main differences between the SOMA data model and data SRAT?
Yeah, sure. So one important difference with ANDATA is that SOMA supports multimodal data sets. So you can have multiple SOMA measurements in a SOMA experiment, as long as each of those measurements was produced from the same set of cells, which are indexed in that top level OBS data frame. But like ANDATA, SOMA supports the concept of layers for storing different transformations of the same assay within a measurement. And this is a way it differs from like SRAT v4, which currently limits you to 2 layers per assay along with a filtered scaled version. SOMA gives you the ability to store as many layers as you want, which is more in line with the way things are done in v5.
Okay. And can you also explain a little bit the difference between TileDB-SOMA and SOMA Core with respect to Python?
Yeah, sure. So SOMA is really a technology built on TileDB's stack. So at the bottom of that stack, we have the TileDB core storage and query engine. From there, as I mentioned earlier, we offer many different APIs for different languages and frameworks that people want to use TileDB with. So that would be the next layer. If you're working with SOMA's Python package, we have the TileDB core engine. We have the TileDB Python API on top of that. And then SOMA leverages both of those dependencies to provide all of the Python functionality, along with the more, let's say, biologically oriented semantics that allow you to easily ingest and export data from these common single cell data formats. So if you're just working in some kind of general analysis, outside of single cell data, you have a large collection of CSV files, let's say. TileDB-Py is a perfect fit for that, and we provide functions that allow you to ingest all of that data into an appropriate TileDB array with basically one line of code. And then you can query it using very pandas-like semantics. So it'll feel very, very familiar to you if you're someone who uses pandas a lot in Python. But again, SOMA is really kind of a specialization of these APIs that is specifically geared towards people who are working in computational biology with some kind of omics data.
I think one quick thing that I would add to that in terms of SOMA core. So SOMA core, it's really an abstract API specification. So if anyone wants to come in and develop a version of SOMA that's based on DocDB, they can utilize SOMA core to just implement it on top of DocDB. And so there was a lot of effort put into enabling this open source type of developing community in case anybody wants to come and provide different solutions to SOMA that we currently have.
All right, well, that's all the time we have for today. We'd like to thank our speakers, Pablo García-Nieto and Aaron Wolin, and our sponsor, TileDB. If we didn't have time to get to your question, we'll try to follow up with our experts. And as a reminder, please look out for the survey after you log out to provide us with your feedback. If you missed any part of this webinar, or if you would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.